Today, we're very happy to have a speaker, Danilo Lyra. He's a bioinformatics breeding research at BASF, Ghent, Belgium. He's going to be talking about digital phenotyping and genotype-to-phenotype models to dissect and predict dynamic phenotype in crops. Danilo was a master student, then PhD student in genetics and plant breeding at the University of São Paulo. While he was pursuing the PhD degree, he was a visiting student to Iowa State University. Before we accepted him, we asked him to take a TOEFL, exam. He took it and have a good score. And then he took a Jack Decker's class and took my class. He was always very driven. He finished one paper together with us, and he finished a lot more papers for his PhD research. Then he moved to be a research scientist at United Kingdom from 2018 to 2021, leading several projects in hexaploid wheat. He published many papers. One of the papers we really liked was published in Journal of Experimental Botany. I hope that he's going to cover that paper in details. And then this is going to be a great opportunity for many of our graduate students here to see how Danilo combine this quantitative genetics, genomic selection, high throughput phenotyping, imaging analysis all together to wrap up the nice story. Thank you very much, Danilo. The floor is yours.
All right, let me share here the screen. Oopsie. Can you see it? Yes?
Yes,
oh.
Yes. Mm-hmm.
Okay, good. I'm not sure. No. All right, Jamie, thanks so much for the introduction, or also for the invitation to talk about this publication that we had. And for me, it was a pleasure to work with you. Definitely was one of the most enriching experience that I had in my career. And I had all these learnings, and I keep them with me in secret and use them as I can. And yeah, and I moved to UK right after I finished with you. And in UK, I was quite quickly here our what is being done at Rothamsted Research. And then after finish my training there as well, I came here to BASF in Ghent. It was May last year, and here in BASF, I'm the wheat crop lead, most on research scientist on working experimental designs, data analysis, genomic selection, and many other functions as well. And what I brought for you today, it's structured in three different sections, three main sections, as the digital phenotyping, going through what's going on in the phenotyping of these dynamic traits, and then modeling the longitudinal traits, what we can do when we implement molecular markers together with these growth data sets, and also show a little bit what the publication in JXB. And lastly, talk about very briefly on predictive breeding and how these G2P models have been used in combination of all these phenomics nowadays. Okay. But before that, I want to just mention a few of the UK agriculture research that you might not be that familiar, and it would be nice if there's a good chance for you to know a bit more of the research institutes that we had, the UK have, which is the Roslin Institute. I will not go through much this, but the Roslin Institute, where they create all the clones of the dolly. And in England, we have four main research institutes: NIAB, Rothamsted, Earlham, and John Ennis. And the main source of funding is BBSRC called. And of course, the whole UK is only wheat. You think half of the place that I stay is only wheat. That's why they have this plenty of studies. And I just mentioned that Rothamsted Research is quite non-recognized by the great contributions of Ron Fisher. He worked at Rothamsted for 14 years, and he published the ANOVA model when he was working with all these long-term experimental designs that they had. And of course, he published this main publication, I would say, all this infinitesimal model in 1918. And of course, if you're interested in history, take a look and read this Lady Tasting Tea. It's excellent for combining history with statistical model. And in UK, we had this designing future wheat breeding program, the DFW. It's a coordinated breeding program, I would say, between many research institutes, the universities, and also the private sector. We had this very organized way. It's really nice. And as you can see, we have four main pillars: development of elite, wild relative interaction, blend races, and synthetics development. And of course, supported by these two themes, as they say, phenotyping, genotyping. And the core goal of this program is to deliver to community what they call a breeder's toolkit. They have breeder's toolkit for many other years, but I'm just showing 2020-21, in which they try to bring new materials as new introgressions, new mapping populations that I will link with the one that I work. It's coming from this DFW, Watkins collections, one of the oldest collections in wheat, and also to provide the community available these markers as well, with exome capture, GBS. In the time that I was there, I was trying to target more SNPs in different regions, and that's the core of DFW. And if you all these new materials that they produce in DFW, they bring and store in this website called Seed Store. If you like to, you can order some seeds if you want. You go to this website and order if you like to study a bit more. And I'll just mention these two that I worked, the Chinese Spring by Paragon and the wheat tealing. I'll talk about them a bit in the previous sessions. Okay, going more deep on the topic of discussion today, okay, not sure if you're seeing this top on the top, but just two main pillars again. That's my focus, my research focus: quantitative genetics and statistical genomics. If you look at quantitative genetics in general, is of course the study of the architecture of complex traits. And recently has been published this homogenic model that people are talking about. Of course, it was a few years ago, but it's basically how many, it's all the whole genome is playing a part in the contribution of complex traits. Basically, of course, the main core of quantitative genetics is to study the G by E interaction. And when you move forward to statistical genomics, now we have this multi-omics dissections, all these omics, and of course, all these old studies of PTL mapping, GWAS, gene-based, and also genomic selection. And just to bring you this content, quantitative genetics links the phenotype with the molecular markers. And the main question here is what we're going to do with the quantitative genetics in this era of phenomics. Basically, if you look at the past, we had more phenotypes than markers. And now, of course, we have more markers than phenotypes, and everything could be balanced in the future. And that's the main goal, to develop these new technologies into what we're going to do with that, basically. And I always like to bring this timeline when I have the chance. It's again, science is moving quite fast. And if you look at the timeline, we have this quantitative genetics converged with the Mendelian genetics and the biometrics genetics led by Fisher, as you can see here, all this variance decomposition in the 1920s. And of course, all of these interactions between the molecular genetics and the phenotypes, as you can see. And in the 1994, we had the first QTL mapping publications, and right that we came for these diverse populations of GWAS to increase the resolution. And then 2022-21 came the first idea of genome selection. Jimmy and you published Rex, all this first practical application of genome selection in May in 2006-07. And now, as you can see, everything is merged. So all of the areas is merged nowadays. There's no distinction. You can use all the methodologies applied in humans and animal breeding with this. And also, we have the main core now. It's everybody is working on. It's phenomics. And I'm not talking about all the phenomics, what the cameras, what we are going to do, what you can do with this new resources of remote sensing. But I will mention only this platform that we had in UK at Rosmstad. We had this field scanlizer, which is a fixed platform implemented 2006, 2016, 2015, 15, if I'm not wrong. It's fully automated. That's the main core, and the high resolution that you can get compared to drones or others facilities, it's this. However, of course, it comes, it's a very short space that you can put your trials, and also we try to increase the area, and of course, it's only one place. You cannot have this G by E, and then you have to look at by drones. Of course, we have drones as well. And I just want to mention this when I talk about my publications, just to keep you in mind that we had this. And it was one of the first platforms implemented by Lematech at that time. Of course, companies want to go through more dynamic ways of phenotyping than having fixed platforms. And why using phenomics? My perspective would say it's to reduce and eliminate the manual measurements. I would say this would be the greatest benefit of it. And non-destructive analysis of growth, morphology, studying key growth stages. And we had this discovery of QTLs, the genes for temporarily dynamic phenotypes. That's the core that we had. We had many traits that we could pick all this variation across time. And that's what I'm going to bring next, the phenotyping dynamic. As you can see, we have many different cameras as the RGB, LiDAR, and near-infrared. You can pick different spectra across all of these wavelength and hyperspectral cameras that we can pick and study the traits. However, you can use the very simple basic one, which is the RGB, to measure all your dynamic phenotypes, which is the canopy height, biomass, disease progression, senescence. You can use all of those using the RGB, which is the simplest one available and cheap as well. And that you can have this many information from longitudinal trait. As you can see, this I'll show a bit more of this, our recent publication. And when you think about dynamic traits, as you are dealing with many different time points, of course, you can look at your growth stage and see what's happening in each stage development. Let's say wheat, we have many growth stages. I just partitioned here the vegetative component, the heading, and the grain filling, only 3. But you can have many different growth stages. The growth stages can easily be predicted by using the GDD. But if you're using the technology or the cameras, you can really split very accordingly in this. And I will not talk about the envirotyping, but what we are also interested in the phenomics is combined with the envirotyping, all these environmental covariables to associate as well with the phenological stages as what's happening within each stage. For instance, heading, hot temperatures or extreme occasions of drought or frost, it really can have bad and severe with your trials. And again, what we see, we measure the dynamic phenotypes, we can split it in the growth stages, and then we are talking about how we can use that information. What is that information? That's the main question. What is functional data? What is usually known by the literature is functional data analysis, FDA, what we call. And the FDA is nothing more than highly frequency measurements, repeated observations. And if you think it's a really simple complex, it is not a simple complex. It's a very complex process that you have to deal with. You have this known biological information to deal with in terms of statistical modeling as temperature, precipitation, what's going on with all those values. And we have many challenges, as I said, about when we look at the functional data. We have noise observations, and how we're going to regularize and do the smoothing of all of these parameters in measures of variation, and how to estimate these confidence intervals between these time series data. And again, I'm bringing these terminologies because we have many different terminologies of functional data, as one of the most common is time series data, repeated measures, longitudinal traits, functional valid traits. Sometimes you see I'm speaking, sometimes I speak with all of them at the same time. It's confusing, but they say, to me, they say the same thing. But if you look for the animal breeders, they could say some differences in these terminologies. Okay, and what we're interested in these data, we're trying to model these trajectories using the mathematical functions. That's our main goal, is we can pick the variation using the functions itself. And when we look at distribution of these functions, we are mostly interested in the mean, variance, and covariance. Keep this in mind. Of course, the relationships of functional data to the covariates. Let's say I have environmental covariates. And I'm just plotting here what I have done for canopy height for wheat. We are very looking to model the covariance of these measurements. It's very important to look at this measure in all of these functional mapping approaches, and the correlation as well. As you can see, height is quite known. As we increase your variance and covariance throughout time, and the mean as well. And this is one approach. You model the function, and then you have your to fit the function. Basically, it's around the same, but you fit these curve functions. You have both approaches, parametric and non-parametric approaches. And again, the covariance matrix are very important as you try to find these parsimonies as much as more parsimonious variance covariance matrix throughout time. And I'll just plot in here the most common one, let's say, that people use is the logistic growth. You can estimate these parameters, and with these parameters, let's say you have 3 for logistic growth curve, and you can have more as well, but the traditional is 3. And however, this is a parametric way of dealing with the time series data. Okay, I'm not really expert on this, but looking at all this literature, and then this guy Ramsay, he published many books on the functional data analysis, and of course, it's not a simple complex. As he said, you don't want to make inference about complex processes using parametric assumptions, as we are very limited in terms of parameters. We want to expand on that, and expand on that, we want to accommodate, we want to pick as much as dots in the growth as possible. And that's why the literature and the community are using these much regression splines. Of course, you can use others, but I'm just mentioning one. The non-parametric one is the regression spline, which is the one I used in my publication. So basically, regression spline, you break down all of your data, your time point data into small pieces and get information out of that small pieces. That was they call the knots. And you have this Fourier basis, you have B splines, you have P splines as well, you have many. And the B splines, it's very flexible. I personally like the B splines. If you look at how it works, the splines very, very easy to implement as well, and tells you much about the data. And then you have two properties when you think about regression splines. One is fit the curve, fit the data. And the second is to fit the smoothing penalty. I'll not go through all the details on it, but the smoothing penalty, as you can see, we have 3 scenarios. We have 2 smooth when we have these time points As you can see, fit this growth, this curve. As it's too smooth, you have too rough smoothing patterns going on here. And as you can see, you have very smooth curve, telling much about the projection of the data is, as they say, is just in smooth way, just right. And this is what I can say about the regression splines. Just to finish this session, I would like to say that repeated measure, it's being used for a long time. If you read the papers, it's not this, it has been used in animal science for a long time. And what I want to bring in this paper is one of the properties of time series is, as you can see here, longitudinal experiments, strong positive correlations between successive measurements. As we have closer the measurements, higher is the correlation as expected. And we have this heterogeneity of the variance covariance matrix over time. So they look at all of this perspective in a statistical model. They know how to do. Basically, we're bringing all this information to this to our needs nowadays, especially because we have many time points going on, and that's our challenge. Okay, what I have just said is a very basic perspective on how to model the growth trajectories. You have the means, variance, and covariance. You have regression. You have to fit your growth curve to any kind of mathematical function. You can choose one. Non-parametric functions are quite flexible, quite good to tell you much about your data, the variation of the data. And the second topic here is how we are going to model these longitudinal traits plus the molecular markers. You're trying to find signals. You're trying to find all this variation when you incorporate this biological information. And I will tell about my wheat publication. First question, what is functional mapping? Functional mapping, again, when you incorporate these biological principles of growth into the model, that's what we call functional mapping. However, you see in the literature that functional mapping is also confused with all the annotation of some SNPs. It's a different perspective, but keep in mind that functional mapping is when you are trying to have multiple time points and link with the growth and development. And again, what we're trying to look, as different from the universe, because you have single time points, but you also trying to get more information out of these growth curves. And you can, this type of model can say you many questions about when does the QTL turn on, turn off? What is the duration of the genetic expression of QTL? As I would say, we have this transient QTL, it appears quite quickly and turns off, and the persistent QTL throughout the growth stages and so forth. And I'll just mention this publication, the first publication, one of the first publications of Wu's group in the University of Florida. He was one of the group that developed all of these implementations of functional mapping in crops, I would say. And keep in mind, this was one of this first publication. And what's the workflow? The workflow that he developed, and they recently implemented this in the FunMap2, it's our package for QTL mapping. He also has a new for functional GWAS. He also his group published on this, because if you think of QTL, you can also apply for GWAS. It's the same philosophy, I would say. And the workflow for functional mapping is first, again, you fit your curve and then you model your covariance parameters. It's mandatory for any functional mapping approach. And again, this is the workflow of his package, and he has a selection procedure to find the best curve fit and also the covariance parameters. So it depends, you can pick and choose whatever you want. However, they don't have this much known parametric curves, which I tried to use as well, tried to understand, but was not flexible for me because I had 26 time points. And then you go through, you do all your QTL scanning in the parameters. So basically, when you have too many time points, you have too many parameters to estimate, and it's quite overwhelming. And then you run all your permutations. And then you have your results, all your lot score, and you can measure the QTL effect throughout time. You can see the QTL genotype, and the QTL genotype, it's what I'm talking about next. Wu, he published this in 2006, in which he mentioned one of the first ways to interpret a QTL genotype, 4 different patterns that you can see in the growth. As the first one, as he mentioned, this parallel growth curve in which you have both being expressed throughout the growing season, which they call permanent QTL. And the second he called early QTLs, each expressed at the early stages, and then it turns off at late stages. And then you have the late stage, which is the opposite, the other way around, which don't have many effects in the beginning, but you have this large effect in the end. And you have the inverse QTL genotype for this. It's a way to find out what's going on with your population and the QTL that you found. And I will bring the last property of functional mapping, which is why to use functional mapping itself. Why don't you use only each time point as it is, and that's it? And what is known in the literature that the functional mapping, it has higher statistical power to detect QTL compared to the traditional approaches. As you can see, again, in his publication, you have the functional mapping being detected in more measured in 3 different weeks. As you can see, didn't detect the interval map in this. However, it was detecting the functional mapping. As you can see, each peak could be 1 QTL, but let's say you have this QTL in here. It was not basically even picked using the interval mapping. And yeah, keep this in mind that this it's also a property of functional mapping. Okay. Next, I'll talk about my, I explained the properties and how do you think and what is a functional mapping itself. I hope you understood that. And I would mention my publication, what we had basically the same questions as any functional mapping study should have, as can we identify contrasting QTLs when you use single and functional mapping? If you increase the number of time points, can you increase the power to detect the QTL is one of the questions. What's the behavior of this, again, this QTL for persistent QTLs, the transient QTLs? That was some of the questions that we tried to answer in this study. I just mentioned here, as you can see, wheat breeding only exploits 10% of the genetic diversity. That's why I bring the next slide of wheat. Why study wheat? Why wheat is so exciting, James? Because of this publication, science. Again, we're always trying to find new genes, their annotation, what's their function, and of course, their polyploid. And as you can see, recently it has been published the wheat. The new version of the wheat genome has been 3 years, I would say, not this updated. And then we have around 100,000 genes, high confidence genes. However, we don't know entirely their function. And as you can see here, we have the wheat genome. Basically, what we call is genetic redundancy, as we have 3 copies of the same gene. And this brings all of this hidden variation as we try to explore this hidden variation. Okay, I'm just showing here the main genes found and most used so far in the breeding community. And I will just bring one gene, which is, you might have heard about it, is the RHT gene. Reduce the height. As long as I work with height, I have to understand what are the genes involved in height, plane height, and of course, one day one friend asked me, why are you studying height? Because you already know all the genes involved in height, and why you keep studying that, and so on. And, okay, yeah, you're right, totally right about that. However, we still want to study more about the genetic variation of the alleles known, and also to incorporate new variation that I'll show in the next slide. That was my answer for that. And as you can see, we have the RHT genes. We call the same dwarfing alleles. It reduced the stem elongation, basically it increasing the grain yield. As you can see, we have these tall varieties as you have less grain yield. And in some, depending on the inbred lines, the varieties that you're releasing, again, you have this high redundancy, and depending on the situation, you have some alleles, and some varieties you don't have that alleles, and that it brings this alleles of RHT comes with a high pleiotropic effect as well on seed germination and seed vigor, and also many other small traits that could affect your selling seeds and so on. That's why you have, by the way, yeah, Norman Borlaug, I'll just put it here just to give a quick glance that this was the father of green evolution, you know that, and he worked as well with the RHT gene, so he implemented all these varieties with RHT in Asia, in Africa, and Mexico as well. Okay, as you can see, we have the RHT genes, and we're always looking for novel alleles and to be implemented. It is a picture of Rothamsted. They're always trying to look at the field. What are the new variation, the new combination of alleles that you can have? And that's, again, the explanation why we have to study very studied traits as plant height. Okay, going forward to my study, we use this mapping population Chinese spring by Paragon, 200 individuals, 26 time points. It's quite a lot of time points, so we could play around with this data. It's not a very limited information, and having 26 time points, it's easier, again, as I said, to play around, to pick and choose in a non-smart way, I would say, because we already have the information. And as you can see, how we phenotype this data in the scan analyzer, as I mentioned, we have the 3D cameras. With the 3D cameras, you can have this type of image. And if you have this kind of image, you pick the point clouds and measure we have this pipeline. I will not talk about this much, but in the publication, it's the supplementary material. If you know more how this is done, it's very specifically when you have 3D cameras. We have this picture of 23 days after sowing, 128 and 228 days after sowing. Sometimes it's quite messy. We had some problems of lodging at the end, you see, but we still keep it, the information. Okay, and I will link with what I said at the beginning, how to model, how to understand this data, okay? As we have this variance covariance, how we're going to model it first, at the end, in between, at single stage, two stage, and so on. Our strategy was to first run a mixed model, and we have the blobs, and we run a factor analytic model, FA model, with the variance covariance as AR1. And we have this pattern of growth. You can see in the tillering, the stem elongation, and we have some, again, as you don't know what is noise, could be one question. Noise is this, when you have this balance, not this good estimation with some genotypes in the head and flowering in here. And the second step would be we smooth this. We use the B splines with 8 base, as we have 26 information. You cut in pieces and extract information. That's what the regression spline is doing. And then this third step is to run a functional mapping. You basically take all these smooth B splines and run a functional it's a PCA component in the end, PCA analysis. And then you basically reduce all of these parameters to 3 or 4 components. This analysis, this workflow, it has been published by Kwak and Carl Broman at Wisconsin. That was his publication in 2016. He had a publication in 2014 when he didn't use the splines. He used the traits directly. However, when you have less than 5, you can use directly the phenotype. However, if you have more than 5 or more than 8, I would say as well, you can fit the curve, you can choose whatever you want. And if you want to even reduce even more, you can apply the functional, the PCA. And then the PCA is your final value. And in his publication, we have this S-LOD, M-LOD, and HK-LOD. S-LOD treats, if you look, each time point independently, okay? And then he takes the average in a rough way, I would say. And the M-LOD is the maximum. You treat each time point individually and take the maximum. It's your M-LOD score approach. And then HK-LOD is your multi-trait approach. Your multi-trait approach, you take your PCs, and that's your final value in a multi-trait QTL mapping. The results of our 26 time points and 2,000 markers, we run the interval mapping, the weakest approach, the composite interval mapping, the fairly good approach in the multiple functional mapping, the more robust approach. And then we try to summarize in one picture what was the results that we had, of course, to understand. As you can see, we can have plenty of information in here. It's something is happening in the growth stage. First, we treat as each time point independently. We treat each time point for running the approaches. As you can see, we have the early stages QTL. Let's say this one, let's consider this one as was interval mapping. We have late stage QTLs as the 7A in the chromosome 7A, and QTLs spread out during the growth season as the 2B. What we call this, when you have more than one time point being detected, we call this persistent QTLs. And when we have only one time point, we call this transient QTL because it's just popping up, popping down. Not sure use English, but it's just disappearing. We don't know what if they're real true or false positives and so on. But we studied a little bit deep on that. We went a little bit on this interpretation, as many publications already know about these transient signals. And we compare with the functional QTL mapping. As you can see, we have the QTL mapping being detected the same here, but didn't detect this. Didn't pick this one, pick this one, pick this one, didn't pick this one. As you can see, the transient is quite hard for them to detect. And it picked the one that was not detected in any single interval mapping. And it detected an additional QTL that was not detected in using the common univariate approach. Okay, that's the main. And then we can go through deep on this persistent and transient, and we run some simulations. And again, as I said, we can pick and choose, we can play with the data. And what we call this, I've just mentioned the systematic variation. You pick the time points as more spaced as possible to pick the whole variation in the growth curve. As you can see, we picking the beginning, 2 in the tailoring, 3 in the tailoring, and simulation. We had more different ways to pick this, pick and choose. Again, it was not a smart way, it was just to play around the data. And we simulated this, having again, this is for persistent. We simulate when we increase the variance explained of the QTL, and we observed a slightly increase in the statistical power for functional QTL mapping, but it was not this much, but indeed was a little bit more. And we used all the approaches that I mentioned for 5, 10, and 26 time points. And we didn't see much difference between the time points. However, when we looked at the transient information, QTL, we observed that the interval map for the individual time point, it has higher statistical power. As I said here, we didn't pick for functional QTL mapping this QTL, neither this and this one, and it was shown here in our simulation that if you have many data points, possibly it's diluting the effect of it, and you're not picking that with the functional QTL mapping. It's one of the explanation, and that's why what we generally recommend, and not us, many papers, well, use both. They're simply easy to implement, as they are suited for different time frames. I would say shorter and long time frames. That's our recommendation in this. Okay, this was the main message that I want to bring to you in this publication, and I will move forward to our next. At least I started doing this, and then I stopped because I moved to the company, and was to play around in a quantitative genetics way in the Tilling panel. We worked with Cristobal and John Ennis and Andy Phillips at Rothmanstead, how we could do some fun with this ultra rare variants. Tilling population is a mutagenized population. We had around 100,000 genes. All of this information they're available in sample plants, and for cadenza, the hexaploid, we have around 4 million mutations. Okay, and 4 million mutations, and the methodology, I will not explain all of this, but the methodology is just to apply a chemical inducer to happen the mutation, to generate the mutation. It's not this random. It's more on A to G, adenine to guanine mutations. And again, after they run an exome capture in all of this. So they have the mutation, the very specific mutations for each gene in the genome. Okay, and of course, this is mostly done to pick the function of the genes, which is not my area. And then we picked all the mutants inside all of the SIFT score as non-coding, synonymous, and non-synonymous, and we'll try to understand what's going on with these mutations. It's really not a common way to run any association map. It's not even a mapping population or QTL population. And we had this 800 Tilling lines in the field, in the field scan analyzer. And again, sometimes we had issues, as the previous publication, we had only 2 years. And all that model is for 1 year, and the other year was not this good, but still we implemented. And our main goal was to run a quantitative genetics pipeline, as breaking down mutations, running the gene mapping instead of having QTLs, or we have the gene itself, we have the combination of variants inside the gene. And then our main purpose was to look at this gene dynamics in the plant development. And as you can see, we had the same strategy looking for persistent genes, transient genes in a very specific way of the mutations, breaking down mutations. And just to show here is a very preliminary result. So we stopped on this. We might get back. We still have more results. Just to show that indeed we have many patterns of persistent genes. As you can see here, this non-synonymous is more present in the heading. It's the way that we observed in synonymous. However, we observed this non-coding in the stem elongation. Why? We still need to look at that. And finally, it was to see the proportional variance explained throughout the growing season when we break down all of these three components, and we observe the opposite, saying that the late stage have more variance explained for the non-coding mutations. But it's a way how you play with the time points, time series data in more genetics way. Okay, this was just to bring this knowledge. Okay, I think it's too much information in your head, but I still have to talk very quickly. These will not be intense of predictive breeding, and we also run some predictions in that mapping population. And when you look at the predictive breeding, what researchers are doing, they're linking predictive breeding with the genotype-to-phenotype models, the G2P models. And what they're trying to do is incorporate the secondary traits to improve yield prediction, as a primary trait is yield prediction, is yield. And also, if you have longitudinal traits, how you can do it? Let's say I have plant height, how is it possible to increase the predictive ability with these longitudinal traits or not? And just mention in some pictures in our, there's a chapter that's coming next month, I guess. And I'll show, basically, we did this literature review on some models when you use digital phenotyping together with predictive models. Okay, the first aspect of it is to use secondary traits. What are secondary traits? Secondary traits could be any traits that are not the primary, logical, no? But it could be any, could be the whole hyperspectral information. It's a secondary trait. Could be any other flowering time, could be vegetation indices, and those are considered as secondary traits. And again, you first go to the field, measure with remote sensing cameras, your population, and then you might have this, if you're using hyperspectral, okay, let's say you're using this, and then you have all these wavelength. I think you have from, this is not the wavelength of the hyperspectral, it's go more to 2000 at the beginning I showed, but you have this raw whole wavelength that is picking biological information mainly. What's in sugar, what's protein, we don't know what's going on basically in this hyperspectral information. However, you can estimate the vegetation index as they're easy to interpret what's going on, and you're picking what is reflecting as, let's say, senescence, you are reflecting at the end, late stage, it reflects more red. So you pick more of that, and it's easier to interpret the result. Okay, so you have your core raw, and then you estimate the vegetation indices. In the vegetation indices, you have plus more than 100. It's quite a large amount of vegetation indices that you can have. And the literature are very interested in the vegetation indices, implementing them as covariate or together as multi-trait and so on. And again, you have your target, your primary target, I would say yield. That's, I would say, the main goal. And your secondary traits, as I explained, I'm just example here in the chapter, is a near infrared spectrum. And what you can do with that, you can deal with many ways. That's the thing. Genome prediction, as we know, it's a single kernel. You just adapt the markers, and when you use the spectrum or the vegetation indices, it's called phenomic prediction. It's already this terminology going on called phenomic prediction, in which you use only the spectral information. However, some publications, they're saying that we are not picking in genetic component, how we're going to explain that in a real plant breeding program. It's complicated. But anyhow, you can combine both if you have the resource and so on. You can combine both. Multi-trait analysis, again, you have your target combined with your vegetation indices, and mega LM is this new approach that Runcie is trying to bring in other in other approaches, selection indices, factor regression, and crop growth model also plays into here, and so on. Shu, in his classical publication of the environment type in 2016, he brought this 4-dimensional profile. It's really in 2016. Yeah, we seen this kind of information now. We have the environment that we don't know what's going on with the environment, and we're trying to bring now the 4th dimension with time series. I would say, in a practical point of view, it's really crazy, but that he's bringing his knowledge, how we could use this in a real practical way. And of course, breeding, it's all about environments. So we have to model this by environment. And how we can use this, again, we have to convert the secondary phenotyping data into the genotypical variables, responses, and use them as for prediction for my traits. What are the problems associated with it? Again, it comes as time series. Is it needed to do feature selection or not? And then how do we do? How is the correlation between the traits and many other questions? Again, it's a complex process. It's not easy to interpret what's going on inside it. It requires adjustment and smoothing of these multiple dimensions. And there's no standard analysis. As I said here, you can use how many different approaches as you like, and there's no real best in common sense. Okay, just bring this from this classical paper again of Freddie in Groningen here in the Netherlands. He tries to stress that the experimental design is very important to any breeding program. We know that, but still he stressed on using the spatial trends, spatial variation. Again, if you're using phenomics, you have to do all this feature extraction, and this could be time consuming. And we don't know, really don't know what's going on in the methodologies, machine learning, what they're doing with this characterization, but it could take time. And especially if you have many environments in a breeding program, you can have plenty of entries and in environments as well. And he designed this workflow of levels in his publications. It's very classical. And after have this feature extraction, correcting for spatial trends, you, again, as I said, you model your time series data using any functions that you like, splines and so forth. And then you have to model your environment. What he's saying that you have the environment come to play, you have to have time-specific, environment-specific information, and then you have your final goal, which is yield. Okay, this is also picture in our chapter. We have yield as main trait. However, if you want to predict senescence, canopy height, you can do so. And what kind of model that you can use? And that's the main thing. And also, if can I use senescence as together with yield? Yeah, it's a secondary trait in this case. As again, you can have many different models. The most common ones are multiple trait and random regression. If you look at the literature, you have random regression as cited many times, and multiple trait as well. As in my understanding, sometimes a multi-trait, you don't model any growth curve. Okay, you just use, let's say, vegetation indices. You have, let's say, your vegetation indices, and then you have yield. You just put both together and run your multi-trait model. However, when you run the regression, you can implement the biological curve of it as fixed effect. It's the main difference between both. I'm just bringing the methodologies as just for you to know more about it. And coming back to our publication, just show 2 slides about it that we also run for the canopy height. And as you can see, we run the factor analytic model and the GBLUP model. And as expected, you have more genetic variance for when you have late stages, especially in the heading. The factor analytic model as expected, you have more variation around the late stage than the genetic component, the GBLUP, when you add markers. And you can see the late stage, we had this lodging, but still we kept that, and that's a decision that we had to discuss and just kept that. And again, the heritability, it's a normal way to do what's the heritability across time. You have higher heritability at late stage. That's what you can find in the literature and other studies. And higher the heritability, you can have higher predictive ability as well. As you can see here, predicting each time point specifically, you can have higher predictive ability at late stage. Just consider the last one. Okay, and then again, we had many questions. We had 26 time points. We could play around in a smart way, non-smart way, and we used the same workflow as the functional QTL mapping. We brought that information. We can smooth. We can have functional PCA, and the first question that we had is, how often do we need to measure traits to improve prediction? I don't know. Can I go to the tailoring? Is it needed to have 26 and the drones 5? Okay, the researchers don't do this. Of course, they go 5 times or 10 times for studies in the academia. But is it needed to go through really at the beginning of the stage for picking QTLs or prediction? They both have different purpose. If you're looking at the genetic signal, we encourage you to pick as many traits as possible, of course, because you have the time-specific QTLs. If you have that curve, even more, you could have more information as well. But for genome prediction, there's no need. There's no need for that. And as you can see in our results, look at the individual time point. Let's say the highest one was 36.36. And when we use the 5 time points, 10 and 26, using the strategy of the growth stage. What is the growth stage? Picking the measurements at late stages, because we know that at late stage you have higher genetic variance, and there is no need to go before that. And if you pick the ones that are higher after in the heading and after heading the ripening stage, and we use the coefficients of the splines, basically. As you can see, we have 3 for it, 4, and here you have 8. But that's it. I mean, it increased the predictive ability when you use that coefficients. I mean, that's the best strategy. You can use random regression if you want, or a multi-trait regression if you like as well, for training purpose, publications, if you like. We had the senescence as well. We didn't have the time to work with senescence, but one day we go through that. And okay, 5 time points collected at late stages are representative. That's our conclusion. And then again, we have this only for a single environment, single year. It was just Benjamin sent me the. Can you present? Yeah, and then I had to go to the literature to see what's going on, what's new about it. And then I found this from Fred. And what he's stressing is for wheat, he measured for canopy height in many different regions, Germany, France, as the really different microregions. And one way you can work with it is have this first and second derivative, and for each environment, I only use one, but you must have for this genotype at time point environment specific. That's the final goal that he presented in his publication, and he's keep going with this methodology, and that's it. If you want to work with multiple time points, that's the goal in breeding. Okay, almost getting there. Take-home message: integrating genomic and phenomics data will unlock the hidden variation of crops, not only wheat. You can work with many crops as you like, but it's still encouraged to combine both as we have plenty of different genomic resources now. We had works, collaborations for metabolomics, gene expression. You can play around that as well. Gene expression, again, we know we take time points in gene expression to also answer many questions. And understanding these functional value traits will help accelerate crop improvement. If we look at the perspective of prediction, we can indeed take advantage of all of this information available. It's again, it has been, let's say, 10 years that we have been developing new strategies of technologies, and it's been working. It's been hard work for everybody. And yeah, high dimensional genotyping and phenotyping data with computational efficiency, that's the word, because we would like to see really efficient way when we have plenty of data as this big data. Is the focus of the novel statistical methods for the next year. If you're enthusiastic researcher, that's what the community is looking for as well. I'm using all these slides from Losmstead, and that's where my connections, and Gancho was my previous advisor. Michael and Nicholas was the head of phenomics. I had collaborations with the microbiology and the physiologists there as well. And to finish that, I could not bring this. This war is getting even worse here in Europe. We are following the news all day, and I work with the Ukrainian British service there, and of course, with all the British service, they had to move, and I heard that one of them it's coming to Ghent to stay a few days to bring his family, but he has to go back. And as I heard, he will not go to the front, but he indeed has to go and help and defend his country. Yeah, that's it. I have. Thanks so much.
Great for the wonderful presentation. Yeah, you're a dedicated scientist to share your insights with us. That's wonderful.
Thank you.
Can you briefly unshare the screen so we can see everyone have questions to ask?
Yes, stop share.
Yeah, clap your hand. Yeah, we still have 21 dedicated scientists to stay around. Go ahead and ask questions. Yeah.
I can share the slides if you want as well, Jamie. Not sure if you.
Just a second. Let's see. We're gonna ask questions because otherwise I cannot see them. Right? Kevin's gonna start.
Absolutely. Thanks, Danilo. I've been sitting where you sat a few years ago, but so I'm just being introduced to the idea, of ETL mapping and session mapping in depth. And I'm wondering, when you say functional mapping, and you mentioned biological, data that you're putting in, what exactly is the biological data that you're adding into your model?
Yeah, exactly. It's what's they're picking in the functional mapping is the parameters. Okay, you have your growth, and you're picking your parameters that are representing the growth of your data. Let's say if the growth is going up or senescence going down, basically you're picking that representation of the curve of each genotype. That's why I show the QTL of each genotype. Sometimes it can answer you what's going on in your growth curve. That's why you bring biology, because you're bringing, let's say, if you have trees evaluated for 10 years, and what's going on with that growing biology of that tree. And that's why you're trying to bring.
Okay. Thank you. I think maybe a follow-up before Boris. I think Boris wants to ask a question. Okay. In regards to limitations, because I think these methods are developed to cover for the limitation of the others, interval and combine, and then you have functional genomics. So what are we looking at as a limitation to this, for example, functional?
Yeah, indeed. I mean, if you look at the traditional publication of Ma, he had all of these growth curves. First, you have to fit the growth curve, and he had the the parameters. As long as you increase the time points, you have more parameters to be estimated. Again, if you have more than 20, I had problems. It takes loads of time to run the likelihood ratios and the permutations as well. And the other limitation that they say that's why Quack he developed that strategy of going first to smooth the data and go dimensional reduction. That one way, and the other way is that most of the methodology already available is only within interval mapping. So interval mapping, we know that they're not implementing having these covariates or covariables with the composite interval mapping is taking. And he implements the multiple QTL mapping just to have more information on pleiotropic effects or epistatic effect information as well. And that's in, if you go to his publication, you see he's saying that why he developed this. And of course, if you go deep on the literature, you have the Bayesian approach, you have machine learning approach, many approaches going on with the functional mapping. And if you go through the GWAS, I really haven't followed what's going on with the GWAS now, but it looks like it's the same methodology. No? The multi-trait model for GWAS, I'm not sure if it's widely adapted, not sure if people are using this much and how much extra information is helping the model as well. Again, it takes time. If you run functional QTL map for GWAS, let's say, oh, it could be painful.
Thank you.
Hi, Danilo.
Hello.
Yeah, this is Boris, and thank you so much for this great presentation.
You're welcome.
And upfront, I would say my research involves functional mapping as well. Yeah, and I've been studying your paper. So,
okay, nice.
Yeah, it's a great honor to interact with you here. So that's great.
Okay, nice.
Yeah. So I was just as you were presenting and having read your paper also, so you use AR1, autoregressive one.
Yeah.
So my understanding was that you need to have equal interval, right, between time points to use AR1. That's where it's most appropriate. So was that the case for your data, or how did you choose AR1? So,
yeah. I mean, the first time when I was looking at the what's functional mapping, I was learning as well. And the first thing is you have to model the variance and covariance, your time points.
Sure.
And again, they are more correlated for close time points.
Mm-hmm.
And RA1 is one of the most used corrections. You can use splines, pre-splines, but I personally like this for a mixed model approach. And again, we had to go through the BLUPS mixed model for correcting the variance covariance. However, we modeled this for the 26 time points. We run one mixed model. We didn't want to break 5. I mean, we could do that, but the reviewers didn't say anything, so we kept with 26 time points basically, which could bring other results, but that was okay. And then having this very sparse collected information and so on, the AR1 will, at least what I know, will do the job quite nicely. We didn't need to go for the autoregressive order to test, because if you look at the FunMap2R package, you can test and see the AIC values of your model, BIC, and see which one is the best. But again, it's how much you'd like to go through all this. It could do. I tried many.
And this following question is just for specific detail. So having explored the FunQTL mapping, I mean, R package, I was just wondering, did you use the FDPCA function in that package, or the normal principal component, I mean, in base R?
You mean the full map package from Quack?
FunQTL, the one, the one.
The FunQTL.
Yeah.
Wait, FunQTL is the traditional one.
Yes, exactly.
Okay, the traditional one. Yeah.
Yeah.
I used that one. I tried to use that one, and even I tried many, but it was not running. It was having many bugs. It was one of the first time that the group was implementing that. We exchanged some emails, but still it was quite complicated. And indeed, in my situation, the Quack methodology was the best way to work around. And that time, many other papers were using and also explain the limitations of the functional QTL mapping. And I've heard from him, he's trying to improve the package now with this non-parametric curves, and improve the efficiency. Yeah.
Thanks. Yeah, my other question would be now coming to the transient QTL. So did you get a chance to follow up, like to check if there is any candidate gene in those regions where you found those QTL hits for the transient one?
Yeah, good question. I mean, we didn't go through this. That's why the QTL mapping is always these open questions, give you more open questions. But one of the questions that we had, where is the RHT gene? Because we didn't pick that, and it definitely was one of the genes that are in major presence in populations. And of course, we didn't pick that because it was not segregating the populations, the parents. So we didn't have this, and we didn't detect. And it was painful to find any association of other genes, what could be going on. It's one of the drawbacks of QTL mapping as well.
Okay, she's raising her hand. She, go ahead.
Hey, Danilo.
Hello, Chi. How are you
Nice to
doing?
see you.
Nice to see you.
It's a really excellent talk. I enjoyed a lot.
No, thanks so much. It was really brainstorming of many things.
Yeah, it looks like you prepared very well, and then have done a lot of scientific research in the past few years.
Yeah, I was just reading some papers to refresh the mind. Everything was organized in my folders, and I had to reopen all of them and prepare and put in. I had some slides already done, but I have to do many new things.
Yeah, nice. So yeah, my question is, when you were explaining the functional mapping, you said it was picking up the curves. So in your understanding, what's the difference between if I just use a curve parameters to do like composite interval mapping instead of doing functional mapping? What's the difference like between these two methods? Have you thought about it, or?
Yeah, you mean if you pick them individually, or if you put in a single stage, put everybody together? Is that it?
Put everybody together, like if you're using your B splines curve parameters, like the ABC that from the fitted curve, and use those, extracting those values for each individual, and then do a composite interval mapping.
Yes, that's the way it's done. I mean, if you don't go through the functional PC, you'll work with these values individually, I would say. But the way of Quack publication, he works each one that individually, each spline, each coefficient that individually, and he takes that S log, that's the average of it. It's very simple. And the maximum, he has his explanation. He wrote his explanation. I don't remember now. That each one is picking a different pattern of the curves. But yeah, it's always to go deep in this and explore. I've seen that you work with this. I mean, in a publication, a recent publication, I've seen that.
Yeah.
Right. In new with phytologists. Yeah.
Right.
For height as well.
Yes, I use a logistic regression.
Yeah, I've seen.
Which worked fine for our case, and I use parameters, which representing one is a maximum growth rate and the other is the inflection
point. Yeah.
Point. But we use a date, like inflection date.
Yeah.
And use that to do the mapping, and that's capturing the curve parameter, like
parameters,
the overall as well.
So have you tried to use others?
No, I looked the functional mapping, but I haven't really implemented and haven't fully understand it yet.
And I mean, for the other curves, I mean, going for P splines or B splines, have you tried to use those, or just go for the logistic unit?
Yeah, the logistic worked fine, so I went with that because I think the P spline or other splines, it's more flexible, and I was a little bit afraid of overfitting, so I think logistic is better because it worked.
Yeah, sure. Yeah, it has been doing the job quite nicely. That's
yes, yeah.
Okay, nice. Yeah, I'll give more insight inside that publication. I just read it, very skimming it.
Oh, if you like.
Nice. No, yeah. Science is science.
Okay, sounds good. Thank you so much. Nice to
welcome.
see you
Yeah.
again.
Yeah, how are you doing? Everything great? Everything good?
Yes, everything great.
Nice.
Having a baby now.
A baby now. How old is he? He or she?
Sorry.
He or she?
He. Yeah.
He. How old is it?
5 months.
5 months. Oh, it's quite new. Yeah. Congrats.
Yeah, thanks. Yeah.
I'm not yet that. In a few years.
Yep. Okay. I'll leave time for other people to ask questions. Really nice to see you.
Nice to see you as well.
Okay.
Good, Juan.
Hey, Danilo.
Hello, man.
I'm so happy to see you.
Yeah, me as well.
How are you doing?
Oh, good. Yeah, we could hard together. Me and Juan in Iowa.
I remember those days.
Yeah, see, old times.
I'm actually so happy to see you working in England, working in those places, doing an excellent job that you did. I read your paper, so I'm very happy for you, actually.
Yeah, move around with the science. Try to, we at least we try to be there. Nice. And you, how are you doing?
Very active. Actually, I'm good. I'm doing my PhD right now.
PhD, okay.
Of this group of students.
Okay, nice. I've seen some pictures of you, I think I'm not sure if Twitter or Facebook or something like this. I'm always seeing you in this.
Yeah, it might be both social media.
Yeah.
So I'm actually working also in kind of longitudinal traits. So I'm very interested in what you are doing and what you were doing so far. And I have two questions for you. The first one is when you look at the signal that you find your QTLs and you make this classification between a persistent QTL, a transient QTL. Are you looking at the signal that you pick in the QTL mapping, or are you looking at the effects that you are having, or are you looking to both of them?
Oh, the signal. If the ones I'm picking as significant in the threshold of the permutations, the loud score, is the one I'm using. Yeah.
Mm-hmm.
Because after that, yeah.
But do you also look at how the effect changes through the session or to different time points?
Yeah. You came in the publication. You have the QTL effect. If it's positive or negative, it's negatively impacting the trait or positively impacting the trait. I didn't show. It was basically, you can pick what's going on. In my situation, some QTLs, you can have that impression of the QTL genotype as well, when it's kind of active in the early stages, and you have more effect on the late stages, or the opposite as well. We found some patterns of this. But still, it's not easy to find. Say, if I don't know, if you go for fine mapping, this kind of stuff, what would help you in this, or so on. These QTL effects sometimes they are so little, so small that the variance I explained is also so small that especially for the transient, that we don't know if they're really true.
And do you explore more? Do you want to explore? Well, actually, you're already published that. Exploring more environments. Do you think that that can have an effect on the signal you pick or the effect that you have?
Do you mean the environment?
Yeah.
Yeah, we thought about the environment. We had only one environment, and that's for sure that it's something is going on, environment specific. And also, even though if we have more information on environments, again, as the last slide show, you'd have the growth specific for each environment, and you might have possibly have used the information from different environments in different time points, and how you to manage that. I still have to think about that in the literature, because if you might have information from multiple years, in your case, not sure if you have it, or multiple environments for time points. That's the main challenge, right? And in my case, it was easy because it was only one single year. Yeah, I had the second year, but we didn't go through this extensive modeling. Do you have this in your situation?
Well, actually, I have less time points that you have.
It's okay.
But we have multiple environments. Yeah.
Yeah, that comes complexity, I would say. You have to find a nice way to bring to the literature, to your publication, your goal. Try to bring nice stuff out of it. Yeah. Good challenge. Good luck.
It was really nice to hear from you, Danilo.
Yeah, cheers, man. Good luck.
Thank you.
Hey, Danilo, long time no see, my man.
Oh, hello, friend. Hi.
Nice to see you.
Beleza.
Beleza. Let's talk in Portuguese. No.
Nada.
First of all, nice talk. You are doing such a nice science. I'm happy for you, seriously.
Yeah, cheers.
And I also have a question about the B splines.
Mm-hmm.
I'm looking at your paper right now, the same picture that you show here, from part E to part F, right? So you have the days after sowing, and then you did a PCA, right, to reduce the dimensionality of the data. So I was thinking that on part E, you model those curves as B splines, right? And then when you do a PCA based on eigenvalue decomposition, you kind of assume that the relationship of your B splines are linear. So my question is this the case, or shouldn't have you consider a nonlinear PCA to go from part E to part F?
Again, that's a good question. No, I haven't even thought about it. That's the thing. And even if I tried, my expectation is that it would not give me this much difference. That's my expectation. But yeah, we have to put it to test. I really not comfortable, or even don't know much about the literature on nonlinear PCAs, but indeed, yeah. And also, they in the Quack approach, they don't model the variance covariance. That's why we did this in the mixed model. And I don't know if they're going to do any implementation. I don't think so, because they published 2016, stopped publishing and working on anything like that. And that's the only methodology that I know that have this flexibility when you have huge information of time points. That's the only I know. Possibly there are others with GWAS again, but QTL mapping, it's very restricted. Yeah.
Makes sense. Well, okay. Thank you very much. It's nice.
Yeah.
You and
yeah. Let's have a chat later. I mean, other times. I hope you're doing well.
Até mais.
Yeah, I have a comment to make for Mateos. That's why I was asking the question whether you use the base principal component in R, or the FGA-PCA in FunQTL package, because I think FGA-PCA does the nonlinear PCA. Yeah, so it's able to pick up. the nonlinear
Okay.
relationship. Yeah.
Okay.
So they
Okay.
have a PCA function in that package that you use. Yeah.
Okay, because the full map from Quack, if I'm not understood, of course, he has many packages inside it, and one of them is the FDA from
Yes.
which I show.
Yeah.
And the FDA is the most complex package to handle functional data analysis, and they basically use that. I mean, if they have more implementations or more variations to work with that, yeah, it's great. You can usually done easily without hard coding in the. It's okay. Nice. Yeah.
Hi, Danilo.
Hello.
Long time no see.
Hello. How are you?
I'm kidding.
Yeah. Yeah. I was trying. I don't know you.
Yeah.
I thought it was Jin Yu, but Jin Yu is where's
Yes.
Jin Yu?
Yeah. Nice to meet you here. Nice talk. I feel you know everyone here. So
Yeah, most of them.
yeah. So I'm a current PhD student of Dr. Yu. I came here in 2017. Yeah, I wish we had overlap.
Yeah, I was close.
Yeah, nice talk. I really appreciate you spend time walking us through all the background and methodology, which is very helpful.
Yeah, thanks.
My question is related to the hidden variation you mentioned in your slide. So you mentioned first earlier you showed a chromosome with QTL and also other part as hidden variation. Then I have been thinking about this concept through the talk. Then at the last slide, that the take-home message you said, so combined genotyping and phenotyping, you can unlock the hidden variation of crops. So I want to, because you mentioned multiple dimensions in your talk, I want to check if I get this correctly. So is it by doing multiple time point mapping, you're able to detect transient QTL? Is that one part of the hidden variation? Then using functional mapping, you increased the mapping power.
Not it relates, I would say, but the hidden variation that was mentioned in that slide, it was published in that I always put the reference in there. It's one publication that the literature review that they say the hidden variation, what they say in wheat is this kind of redundancy that I mentioned, because you have 3 genes in 3 different chromosomes, and sometimes you can have this variation that is not being expressed. I mean, sometimes you have only one gene being out, and sometimes when even if you have 2, what's the combination with these 2, and you have 2 with the other genes, because sometimes it's polygenic combination, right? And that's the hidden variation that I was mentioning. But of course, you can say again, hidden variation you can pick when you have multiple time points. That's one way to view as well. Yeah, that's the core of genetics, not only wheat but other crops as well. The Chilean population, as I mentioned, they are expecting to find the function of the genes using that population to find new genes with that population as well. And they're hoping to, again, as they say, unlock the variation of the wheat using that population. And they do found, I remember when I was in the field evaluating that in the Chilean, and with the research geneticist, and he said that we have so many variation. One of them was when we had all the plants in the field senescence with high senescence, but one was still high green. And he said definitely it's one gene with the vernalization, because wheat we have vernalization, that we need amount of code to flower, and it was still quite green. And it was nice to see that, but it was not good to work with that, because Chilean population, it's a mess. You choose and pick one genotypes that don't grow they have really no balance in the field. When you see plants like this, senescent, not senescent, it's crazy. And yeah.
Yeah. Okay. Thank you.
Thank you.
Go ahead, Yuru.
Hi, Daniel, thanks for your excellent presentation. I
Hello.
still digest your material now, but I have a simple questions about except because in your presentation, you demonstrate the functional mapping in plant height for us, but I'm just wondering which trait in the crops could be used in functional mapping. And the following question is about, is it possible to use in the perennial crops to identify the major QTO contribute to the key trait? Yeah.
The perennial crops, you mean?
Yeah. Yeah.
Coffee, or trees, and yeah, I mean, one of the trials and data sets that they used was with forest trees, and when you have age and growth, yeah, especially growth, it's quite easy to pick that, because senescence, unless you, I don't know how they could work with it, but yes, they do have the stem growth of the trees, and not only crops, but they repeat measures in animals. What they said, they have all this growing with the cows and stuff. It's quite common. Do you work with perennials? That's why you're asking.
Oh no,
Okay.
I work in maize, but I'm just wondering, because from your presentation, you say that the key point to measure the trait were at the late stage. And I just thinking that could be a newer crop, their growth stage is just very short compared with
yeah, exactly. Crop. Only for yearly crops. Yeah, indeed. That's the main core, I would say. For perennials, they do have phenotyping. They do use phenomics, but they have other questions for perennials, especially trees and tree breeding. They have different questions that they want to answer than having this final growing season, I would say, to pick, because they're more interested in the wood.
Okay, thank you.
Okay, thank you.
Nice presentation.
Cheers.
Danilo, follow up the cheese question. I wrote note about your, the analysis pipeline you modeled from Quicken, and that's Carl Burman and Edgar Spalding's group. So adjusted the values and then smooth the B splines, and then ran in the PCA, right? Takes first few PCA. You suggested, take first 2 or 4 PCAs to run analysis. So do you, I was imagining, because I also hear other talks, I was imagining if this curve can be modeled with a simple logistic regression curve, you would generally expect first PCA might be one of the parameters, second PCA might be the second parameters. But in your case, because you have so many data points, 26 of them, so your curve could be better modeled with B spline, which has more segments. You were explaining that, segments, right? Then, but still, if you do a very rough estimate with a simple parametric curve, right, you would imagine that the correlation or the correspondence between your first PCA, second PCA to one of the first 2 major parameters, or if you add more curves, third parameters, there's some correspondence. Has that been shown in the literature, or is that not, like we expected?
Yeah. I was expecting the hard questions coming from Jimmy. Yeah, I cannot comment much on that. I haven't seen this what you just mentioned very specifically in some publications. Really, okay. I haven't, especially because Quack didn't make much comparison with parametric curves It just went through this splines and that's it.
Yeah,
it gave some recommendations.
The smooth B spline is still a matrix of the whole process, and then they do PCA, right?
He uses the basis coefficients. As I said, we had for 26 we had 8 bases. So basically from 8, he reduced even more.
Oh, okay, that's clear.
For 1 or 2, when you have hundreds of it. Let's say when you have 4, you want to reduce even more parameters. So basically when you have 4, I think I had, when you have 5 time points, I think was 2 or 3. And then from that, you have only 1 PC or 2 PCs that you can really account to, let's say. But it's from the basis coefficients, as I remember.
Yeah, I also see that some people, they will just, whatever data you have, they just do a PCA, and then they do the, mapping, and then they call that as a function mapping too, right? Without thinking about the curves, which case you have the longitudinal traits or you have the time series traits. In some cases, they just lump all the traits, do a PCA, and then you map with the PCA axis. Yeah. So that they call that as functional mapping too. So
yeah, I mean, the first publication of Kwak in 2014, he did this. I mean, that's why I said that when you have less than 5, you just, at least he mentioned, go through your adjusted. He didn't mention about blobs and stuff, but just have your values, so 10-point values, and do a, if you want to go through a PC, he went through a PCA, and then use the PCA. But
so another point, yeah. I thought it was interesting that I think if students remember last year we had a Baker symposium. There was one lady from Australian speaker. She actually mentioned about the transient QTL and persistent QTL. We look at her publication, and then I think that publication was probably about the same time as yours. She probably cited your publication. I asked her about this transient persistent QTL, right? And then I identified, oh, was Danilo was the one. Then I think both of you cited one earlier paper about this transient QTL and then persistent QTL. When you were exposed to that, and also you mentioned stage specific QTL. When you were first touching that topic, what was your feeling like? Transient QTL, persistent QTL.
Yeah, again, when we look at the literature, I mentioned the definitions of Wu in his 2006 paper when he had this. He mentioned permanent QTL, early QTLs, late QTLs, and these inverted QTLs. And then, I mean, there's no much on the literature about the definition of transient or persistent. I still have, I've looked at some reading a few days that they mention really persistent QTLs as the main terminology when you have these persistent detections of QTL throughout the growing season. But we had this thinking what name we could call that, especially for the transient. We didn't have a definition of that transient, and of course it's a generalization of the literature. People might be saying different names, or even when you have 2, you're not sure if you can call persistent as well. It's
I think I got literature mixed up. I was talking about transient QTL and permanent QTL. So I actually combined the term you were using with the term that Ronglin Wu was using, because Ronglin Wu, like you said, Ronglin Wu, he was the one that was doing this functional mapping a long, many years. He was using permanent QTL. So of course, my permanent QTL was a mix up from early literature.
Yeah.
And then
we thought about that.
I like your stress about this later stage, right? I gave a presentation, and I think we have an internal discussion. We said, we try to map all the stages, we try to get everything. But the plant in the field, the plant is also integrating all this information, biology, environment together. It's also calculating, and it is showing this final stage phenotype, right? But then, although we say we can do functional mapping, we can discover early stage QTL, I still have the doubt, the question saying that what could have been done more from this early QTLs, from early, this stage, functional mapping. Let's say we almost have to say we want to do a in-season prediction, right? Otherwise, we could just be patient, wait a few more weeks to be the late stage. That is actually not the point I want to make. The point I want to make is that I always feel there's more things we can discover from modeling the early stage, but then we just don't know what's the best way to capture that information.
Yes, that's a really good reflection, I would say. I mean, that's we initially had the same thinking, but again, it's complex how we're going to deal, especially when you have these very, as I showed there, you have very series of QTL detection in the early stages. In the early stages, I mean, really early stages, when you have 23, 50, and 60 days in wheat, and how you can manage them if you, I don't even think about genomic prediction. I really don't know how to connect that with this QTL information, but what researchers are trying to do with prediction is data as well. They're trying to use early stages to the late stages. That's their final goal. Even don't harvest the trials in the field, just leave it there or just not consume. Thinking on prediction, huh? But for the early QTL, what could be the impact of this early in the final? I mean, people could have answered this already. I'm just not. But yeah, it's a really good question. I mean, are you going through this, trying to go deep in this research, or no? In your group?
Oh, we are doing some research, but sometimes we can wait until Boris discovers some result. Sometimes we can think ahead of time. Just imagine what can happen. I think Huang is also doing very critical research in that biomass sorghum too. But the question still being there. We want somebody to discover something to better utilize the information we gathered, right? Yeah.
Yes. I mean, the time that we were discussing this, because we already have the 26 time points. Again, with 26 time points, it's easy. But is there any way that we could find the best time points to measure, to define, not to have random? Okay, is it related to the growth stages? If you look at the specific growth stages, is that the right point for you to pick the QTLs that you want to pick? You just go there and phenotype that specific time. Otherwise, because each year you have different, the growth is different, and it could not detect the same QTLs. Of course, you have this interaction between year and environment. But we thought in developing a model that could, at least for prediction, not for QTL mapping, that would give me the best points to measure in the field.
Ah, that's great.
But at the same point, we've seen that if you look at the late stage, stress in the late stage phenotyping, it's better. I mean, at least this known, the way we have done, there's no need to go back in the really early development. In wheat. I'm not sure about the other crops. I'll definitely not go for that. I would keep you with the late stages, but you could ask this for the late stage. Which late stage I would go? The beginning of the heading, and how many days? I mean, you have a window of 10 days. Let's say in which you have the heading or the flowering, all this 10 days. You can only one time is good, or you can have 2 times in this transition, and then in the ripening, all this grain filling. Does it help? And then you have many problems as well, that is lodging, and you could have for your phenotyping.
Sure.
But yeah, I mean, you can have many questions around it.
Jamie, you have a question? we sorry, I feel like we've been talking.
No, I have learned a lot, but I have a comment.
Sure.
I have no questions. I cannot comment on this, but I have a comment. And I just wanted to say, Danilo, thank you so much. I think if you have not appreciated the enthusiasm from the current crop of students here for you and your research, I wanted to point it out. And I wanted to point out to the other students still on here that like, this is the goal, right? To do such exciting research, you come back and inspire the students that will come after you. So, but as to the research, no, I've just been taking notes and learning.
Yeah, I was really happy about this. When Jamie mentioned that there are people are working on the stuff that you have done, and I was prepared. I'm always prepared for. If I'm going for a talk only with Jamie, I would do the same. Just be focused, be very. I try to bring nice stuff, nice literature.
Okay, let's see. We don't want to drag. Danilo is probably 12 minutes before midnight at his time, right? So he's a very dedicated scientist, right? That's why I encourage everybody to exercise, right? You have to have the energy to do the hard work, right? So anybody have a last question, or should we say hi to, say bye-bye to Danilo? I already take the pictures. Let's all wave to Danilo. Appreciate him one more time. Thank you very much. Thank you.
All right, thanks, Jamie. Thanks, everybody. Nice to see all of you.
Bye-bye.
Bye-bye.